Welcome to part 2 on our video of blood. In part 1, we found out that blood is made up of plasma, which is its matrix, and cells, which are formed elements. Most of these cells are called erythrocytes, which are just red blood cells. And that is this component down here. Now, you also have white blood cells, which are important for immunity, and platelets, which are important for blood clotting. And when you do a blood centrifuge, they make up this buffy coat right here. Now, the plasma is going to be made up of waste products, water, nutrients, sodium and chloride, and proteins. We talked about 3 proteins. Albumin deals with water retention. And it's very important. If it wasn't for albumin, you would lose a lot of fluid. That could lead to pulmonary edema, which would be very bad. It could be edema in your limbs, which crushes nerves. Or in some countries where the infant gets weaned too early and they don't have enough protein, they actually develop a humongous potbelly. I'm not going to try to say the name for this. And it's basically because the infant had no protein in their diet that all the fluid leaks out into their abdominal cavity, which can be very bad as well. So we have 2 types of globular proteins, alpha, which are going to carry steroids, and gamma, which are antibodies. Then we have fibrinogen, which helps blood to clot. On this part, we are going to look at those blood cells in detail. We will match the function of the cell with the cell type and identify unique characteristics. And on the lab exam, you have to identify leukocytes, which are white blood cells. So, erythrocytes, that is red blood cells. They are a biconcave disc, and this enlarges the surface area greatly. So if I were to draw another cell around this red blood cell, that's just a circle. See how much less surface area there is right here than there is by doing this concave surface? So it increases surface area. That is good for diffusion. It is anucleate. It has no nucleus. So it's just hemoglobin. There's no organelles either. So we just have hemoglobin. Red blood cells are a sac of hemoglobin. Now, they're very small. It is the smallest of the cells of your body. There are some debate that sperm are smaller, neither of which is a complete cell, by the way. Now, they must line up through the smallest capillary. This lining up is called Rolex. I like saying Rolex like expensive Swiss watch, but it's Rolex. And again, they're just filled with hemoglobin for gas exchange. So, leukocytes are the fancy way to say white blood cell. They're larger than a red blood cell. So here's an eosinophil. There's this red blood cell next to it. It is humongous. Here is a lymphocyte. It's still about 1.5 times the size of a red blood cell. Here's a basophil. It's 3 times the size of a red blood cell. They are complete cells with nucleuses. In fact, some of them have multiple nucleuses. This guy, it's a neutrophil, 1, 2, 3 lobes to its nucleus. The basophil and eosinophil have these barbell-shaped like nucleuses. So they can even be multinucleated. Technically, it's 1 nucleus with different lobes, though. Now, all leukocytes can leave. So here's a leukocyte getting called into action. Help! We have Bill, the bacteria, getting in. So this helper T cell called for backup. So this neutrophil is going to come and kill Bill. Now, it can lead, so chemotaxis is attracted to chemicals from damaged tissues or other leukocytes. Diapetus, di is 2, pet is feet, means it can walk. It can leave circulation. It's slipping between some epithelial cells right here of the blood vessel, and it is going to go to get help. They could embed in tissues. Macrophages have about 20 different names, okay, maybe 3, because they're embedded in so many different tissues, and they can go to the site of an infection. So, there's 2 general classes These three right here are called granular because I have visible granules. So in my eosinophil here, I have all these nice red granules. In my basophil, I have nice blue granules. Remember the basophils of the pituitary gland were blue also? Then my neutrophil. What do you get when you mix red and blue? That's right, purple. If you look carefully enough, you'll see purple granules in the neutrophil. So basophils, eosinophils, and neutrophils all are granulocytes. They have visible granules. And if you notice, they have multi-lobed nucleuses too. Here's my multi-lobed eosinophil nucleus, my multi-lobed neutrophil nucleus, and my multi-lobed basophil nucleus. Now the agranulocytes are called agranulocytes. They don't have visible granules. These are the monocytes and the lymphocytes. So on the lab exam, I might give you Dr. Spock or Marvin the Martian's blood sample and say you have a granular white blood cell. What category would it fit into? And you would say granulocyte. And I want to also say the nucleus takes up a lot of the cytoplasm in both of them. And it's a single nucleus where these are multinucleated. Neutrophils are multi-lobed. So there is one nucleus there, one nucleus there, one nucleus there. And lazy people, myself included, like saying they're multinucleated. There's really bands connecting them. And sometimes they'll have a fourth nuclei right there, sometimes not. This is the only one that will have more than 2 lobes. That is your neutrophil identification. 3 or 4 lobes. And they're attached by these little fingers. They stain a light purple. It's really hard to see, even on this blown up Photoshop picture that has granulocytes. And if you were to actually look at these on a microscope slide, you'll see mostly red blood cells. Then the most abundant leukocyte is a neutrophil. So neutrophils are first responders. They're attracted to damaged tissue by chemotaxis. I will ask you what chemotaxis is. And they're phagocytes. They're basically Pac-Man. Here is Pac-Man with his multi-lobed nucleus, and he's going to eat Bill the bacteria. So here's Bill. And our neutrophil is going to kill Bill. It also produces chemicals. They're a cytotoxic cell. They release these chemicals in their granules, and these are free radicals. And free radicals are not the hippies left in Berkeley from the 1960s. They're actually chemicals that destroy things. They break double bonds, causing whatever cellular structures have those double bonds to break down. Our second type of granulocyte is an eosinophil. I've also heard eosinophils, and they have these barbell-shaped nucleuses. They also have these brick red granules. Remember the acidophils in the pituitary were red? Well, the eosinophils actually react to the same dye as eosin, and they turn a brick red as well. It's the second most rare white blood cell. And in lab, I always have students look at a histology slide where the person or the rat had eosinophilia, which is an abnormally large amount of eosinophils. And on this slide, we have our eosinophil in action. He is fighting the worm, and he is going to knock out the worm. They respond to parasitic worms, and they release chemicals to help kill the worms. Now, the worm, you could actually see with your naked eye. It's going to be like that. There's a nice wormy. And this is a cell that big. So usually it's just chemicals. The worm is surrounded by a cuticle, and the chemicals digest the cuticle. And it helps the other cells to attack it. Now, when I went to Indonesia, I swear I brought back a parasitic worm. Because whenever I go to the doctor, I have a high eosinophil count. I have 5 per deciliter of blood. That's not very many. Now, it's probably I have allergies, and they play a role in allergies too. But I swear I brought back a worm. Eosinophils are red, and basophils are blue. They have blue granules. One of my colleagues has it in for all of our commercial slides because they look more purple than blue. But they should have these blue granules just like the basophil in the brain. In the pituitary. And there's so many of these that you can't really see the nucleus. So there's really a nucleus in there. Should be a bilobe nucleus. And it is 3 times the size of an erythrocyte. So that should be an M. So they're large. That's the rarest white blood cell. I used to offer 10 points of extra credit if somebody found one. Nobody would ever find them. Now, the thing with them is they are 3 times the size of a red blood cell. A lymphocyte, the nucleus takes up the whole thing, and it's about 1.5 times the size of a. So remember, eosinophils are red, basophils are blue, neutrophils are purple, and I love coffee. Yeah, it's coffee. Basophils make histamine, and histamine attracts other cells by chemotaxis. So on Mr. Spock's blood cell, I might say you have a granular cell that attracts others. So what kind of cell would that be? It would be a granulocyte and a chemotaxic cell. Well, basophils release histamine, and they attract other white blood cells to sites of infection. I learned they were not related to mast cells. There's now a strong bit of evidence that they are related to these things called mast cells, which are what are responsible for the histamines in your eyes and nose giving you allergies. And they attract other cells. They also make this molecule called heparin, and that molecule helps prevent blood clotting. The second most common type of leukocyte is a lymphocyte. Now this is agranular. So that is the cell membrane, and I could see clear cytoplasm. Now the nucleus makes up most of the cytoplasm. So there's only a little sliver. So this is why the students always thought these were basophils when I offered 15 points of extra credit. If you look, it's only a little bit bigger than my red blood cell. Now it's one of the more common ones. It could be up to 40% of a blood sample. And there is actually a granular one out there that my physiology students have found, but we're going to pretend it doesn't exist. Now, there's 2 flavors. There are B lymphocytes that mature in the bone marrow, and T lymphocytes that mature in the thymus. Our third one, our natural killer cell, will also develop in the thymus. They break the rules of being a lymphocyte, though, so we'll talk about them in the lymphatic chapter. Now, they are responsible for acquired immunity. So right now, we have coronavirus going through the population. Right now, nobody has acquired immunity. They haven't been in contact with it. That is why it is called novel. Now, the people that have recovered from it have memory B cells and memory T cells. Those cells will remember the coronavirus, so the next time it rears its ugly little capsid, and the capsule actually is really cute of it, those cells will kill the coronavirus, so they won't have an apparent infection. So this is how vaccines work. You get exposed to a pathogen, and you make B cells and memory T cells to help respond to it. And there's different flavors. There's cytotoxic T cells, there's plasma B cells, and notice our natural killer cell, our bad boy, is being sent off to the side. It's the largest of the lymphocytes, and it could have a kidney bean-shaped nucleus. I don't like this description because there's something that you might see called a band neutrophil that would also have slight kidney bean-shaped one, and they have lots of free cytoplasm in it. Now, monocytes are attracted to damaged tissue, chemotaxis, and they arrive late. They are the garbage disposal unit. They clean up the mess that the basophils left behind. Now, they can also just leave circulation and embed in tissue. They turn into something called macrophages. Do not listen to people saying it's a macrophage. Macrophages are big macro eaters. They eat things. They're garbage disposal units. They clean debris. You have a lot of them in your lymph nodes where they eat fragments of viruses and they'll then report it to